It's always tricky to follow a talk about modernization with my paper notes. But then again, I'm also that person that like reads actual books, like really paper books. I'm that old. So, all right, so I'm here with Dr. Ferrugi, and we're going to be talking to you today about Bio-INDs and comparative analyses. 2 for 1 here. All right, bonus deal. Let's ride. Just as a disclaimer, this presentation reflects the views of the authors and should not be construed to represent the FDA's views or policies. So to start things off with Bio-INDs, I'm going to give you some tips here about how to avoid clinical holds and how to just streamline your Bio-IND processes. So I'm Andrea Dugas. I'm a reviewer in the Division of Clinical Safety and Surveillance. I am a physician, and we are the division that is responsible for reviewing these Bio-INDs from a clinical safety standpoint. Obviously, these are going to be going over to our colleagues in bioequivalence and chemistry and all that, so what I'm going to talk about here really is focused on clinical safety. So there are 2 things that I want you to take away. She just had 1 thing. I've got 2. You guys can do it. I have very high expectations for you. 1 is to be able to identify the FDA resources that are there to assist the Bio-IND process. Hint, they're at the end of this presentation in the resources slide. It's like labeled. And second is to learn some tips on how to avoid clinical holds and save time and back and forth on these Bio-IND submissions. So no FDA talk would be complete without mentioning 21 CFR. This is my obligatory slide. I did it. So as opposed to the Office of New Drugs, where INDs are really going to be a lot of times new to human or not approved drugs, these are already approved. They've already been in humans. So there are 3 reasons why people would need to submit a bioequivalence IND. And that is if the study includes a radioactive product, if it includes a cytotoxic drug product, or if the study has a product in it, and it could be the drug, it could be some other product that is being given above the dose that is recommended on the labeling. And when you submit those Bio-INDs, like I said, we do the clinical safety review in addition to the multiple other colleagues we have at the FDA that do reviews of Bio-INDs. And from our perspective, we can issue a clinical hold, which basically stops the study and does not let it continue, or does not let it begin if it hasn't begun yet. Or we can give you non-hold comments. These are helpful suggestions on kind of how to improve the safety or study design. There are 4 reasons from a clinical safety perspective that we can put that Bio-IND on clinical hold. So the first is if human subjects are going to be exposed to an unreasonable or significant risk. That's actually the most common one. That's the main reason in this day and age that we tend to put Bio-INDs on clinical hold. The other 3, you can are fairly easily avoided with some due diligence. So 1 is if the clinical investigators are not qualified to conduct the Bio-IND. That's rarely the case, but what happens more often is people just forget to submit the CVs of those investigators showing that they are qualified to do that. So that one can be avoided for the most part by submitting an appropriate CV showing that the investigators are qualified. Number 3 is that the investigator brochure is misleading, erroneous, or incomplete. Again, submitting a complete, accurate investigator's brochure, boom, knock that one out. And then the 4th one is that if the Bio-IND doesn't contain sufficient information to demonstrate to assess the risks of safety to the subjects, and again, submit a complete study protocol. So if you submit those 3 things, a clinical investigator CV showing they're qualified, a complete and accurate investigator's brochure, and a study protocol, that's really going to narrow this mostly down to the main reason we put studies on clinical hold is significant risk or injury to the subjects. So we took Bio-INDs that have been reviewed over about 18 months and looked at all of the comments that we gave back to the applicants or sponsors and categorized them into kind of main class categories to see where we are having most of our comments and critiques on these Bio-INDs. And this is a lovely pie chart, even though in my PhD they told us never to use a pie chart. I am a rebel. So as you can see, this little like wedge that's like 8% for application completeness, we already talked about that. That's where you submit the investigator's brochure, the complete protocol, the investigator CVs, boom, taken care of that one. Then, the biggest wedge there in red is selection of subjects, and that's really where we have the most comments going back to the applicants. And so I'm just going to walk right around that pie circle, and we're going to talk about each one of these topics and give some specific tips on how to improve submissions. So, study design and protocol tips. You're going to hear me repeat this over and over again with every slide. My first 2 points are going to be look at the PSG and look at the RLD label. So the FDA issues these product-specific guidances for generic drug development. They are not mandatory, they are guidances, and you can absolutely use an alternative approach, but I would suggest you to give adequate justification. But these are our best thoughts on how to complete these studies, and so they bear to look at and consider. And then the next is to look at the RLD labeling. That's what we consider too. It really highlights some of the safety issues that are going to be occurring with this drug. So, subject selections. Shockingly, I'm going to say follow the PSG. Who would have expected that? But the PSG is going to give you tips on study design. It will let you know, maybe this is recommended more in patients versus healthy subjects. The RLD labeling is going to let you know who should not be receiving this drug, who has contraindications to it, what are some potential drug interactions. So, if a drug is contraindicated to be given to patients with thyroid disease in the subject selection, we as reviewers are going to be looking, make sure patients with thyroid disease are not, are excluded from that population so that we know that we're keeping them safe. Something that I see a lot of is that, protocols will say that they are enrolling healthy subjects with normal vital signs or with clinically insignificant. That is very vague. It is hard for me as a physician to know what you mean by a clinically insignificant vital sign. And so who's ever enrolling your patients is going to have a hard time too. So that's why we really recommend giving specific parameters. Instead of, normal vital signs, say, normal vital signs, and this is what we consider normal. And the same with lab values. With normal laboratory values, okay, just give us your normal laboratory values so we know what you're talking about. If a drug is contraindicated, say in people with liver disease, then again, I will look to the inclusion-exclusion criteria to make sure that you're only enrolling people with normal liver function. And normal liver function means normal liver function within the range of normal liver function that you gave me, not 5% above that or clinically insignificant, liver values. Study drug administration. Again, the PSG will give you the kind of recommended dose. Sometimes they will comment on timing. In terms of the RLD labeling, they'll also give instructions. If the RLD labeling goes into detail on how a drug should be prepared, dilute it like this and infuse it over this time with or without a filter, I would expect that also to be in protocol so that we know that the subjects in the study are getting the drug the way that is recommended be administered when it is given in patients. Also, if a drug can cause anaphylaxis or some sort of severe allergic reaction, it's important for us to know from a patient safety perspective, how are you assessing for that? How are you checking those patients? How often are those subjects? How often are you checking them? Do you have a code card available? Do you have epinephrine available? How are you going to be identifying and monitoring these subjects for anaphylaxis? And then the last thing I'll say is that if you are enrolling patients that are taking this drug for a disease, then for their safety and for their treatment of their disease, they need to stay on their regular treatment schedule. And that treatment schedule should not be changed because of the study. So sometimes we'll see, a drug dose may be delayed for administrative reasons. Well, that's really interfering with the patient's treatment. If the patient needs to stop treatment because of some adverse event, that's very different rather than just for the study. All right, safety monitoring. Again, look at the PSG recommendations, look at the RLD labeling. That will give you a lot of information about the types of adverse reactions that need to be monitored. Again, define what is a normal vital sign range, define what is an acceptable laboratory range. And then consider the timing and the frequency of how you might do that laboratory testing or vital signs or EKGs, et cetera, whatever you're using to monitor these subjects. So if a drug can cause neutropenia about a week after giving it, checking the white blood cell count the next day isn't going to detect that adverse event. That's not going to help protect the subject's safety. You need to check for the neutropenia a week after, and whether that's having them come back for another study visit or combining it with another blood draw. But the timing of it needs to make sense in the context of the adverse reaction that you're looking for. Same will be, if a drug can cause liver injury, then it would make sense to check and make sure that the patient does not have liver injury before continuing to give them additional multiple doses of that. And the one safety monitoring timing that I see actually the most is that, so for drugs with embryo-fetal toxicity, people are testing for pregnancy before each dose. That's great, wonderful, spot on. But also just remember to check for pregnancy at the end of the study, just so that we can, record any sort of adverse events and discuss briefly how you plan to follow that up. All right, subject withdrawal. If a subject develops a contraindication to the drug during the study, maybe that's an allergic reaction, maybe that's a kidney injury, something like that, then they should not be receiving further doses to it. So if they develop a contraindication, that needs to be listed in the subject withdrawal criteria so we know that those subjects will be safely withdrawn and not additionally dosed with the medication. Adverse event reporting. This is actually usually done pretty well. There's a really nice guidance for industry that the FDA put out that talks about safety assessments. The one point in here that I do kind of notice is there are specific specific adverse event reporting for Bio-INDs. They're going to be submitted for marketing for the FDA, that you need to report those to the FDA, and with a timeline. So if there is a serious and unexpected suspected adverse reaction, that needs to be reported to the FDA within 15 calendar days. And if there is an unexpected fatal or life-threatening suspected adverse reaction, that needs to be reported to the FDA within 7 days. And just make sure that that is also included in your adverse event reporting. Informed consent. Again, the FDA has some nice guidance here on informed consent guidelines. The main thing is just making sure that the informed consent is clear and understandable. If you tell a patient that they could have hepatic toxicity, they probably are going to have no idea what you're talking about. Stating as, you could have damage to the liver or liver injury, in a way that is understandable to the subjects. Make sure to fully inform the subjects about the study risks. One tip there is if the label has a medication guide on it, that can be tacked on to an informed consent, and that will walk through at least some of the safety risks related to the drug in a way that patients or subjects can understand. And then also, if you already have a study event table in the protocol, that is something that could be added to informed consent as well, in order to help fully inform the subjects about the study events. And my last point here is just, if there are specific study restrictions that the subjects need to know about, put that in the informed consent. That way they have been clearly informed of them, and we know they've been clearly informed of them. The most common one there would be contraception. If they're required to use contraception during the study and for 4 months afterwards, just make sure that's clearly spelled out in the informed consent so that that is highlighted for the subjects and they know about that. Everything I've said before is about Bio-IND submissions, mostly about new submissions, but for Bio-IND amendments, just to touch base there, if you are amending the Bio-IND, just include a track changes protocol, and ideally it would be very nice to have like a little table with the differences between the new submitted protocol and the current study protocol. That just highlights for us what the changes are, the rationale for the changes, and really helps us kind of understand the thought process and ensure that there is no change to patient safety with this new amended protocol. So this is that big highlighted resources page I promised at the very beginning. Here's a list of a whole bunch of resources. I have those FDA guidances that talk about informed consent, that talk about adverse event reporting. There's information here about when to submit a Bio-IND or exceptions for a Bio-IND submission, and a reference actually to a presentation here 2 years ago that was really great, that kind of walked through the submission of INDs and when to submit it. All right, challenge question. Which of the following statements about Bio-INDs is not true? I will just let you read through these for a second. And in the interest of time, because Dr. Ferrucci told me that if I went over my time, I have to bake her a cake. And I don't have time today to bake a cake for her. So you're going to lose your cake. I'm going to jump. The answer here is bioequivalent study in healthy subjects absolutely needs to consider the RLDs, labels, warnings, and precautions. That's how we, that's how you're going to make sure that these subjects are not having, significant adverse events and that they are staying safe for this study. So in summary, the FDA has multiple resources to help you with these Bio-IND submissions. I strongly encourage you to come back to this presentation online and go to that resources page to find a large number of them. And just again, refer to the RLD label and to the PSG when you're designing your Bio-IND studies. And with that, and 30 seconds over and a cake I have to make later, I pass stuff over to Dr. Ferrucci.
Thank you.